As we know, seroma is a serous fluid collection under the skin flaps post-mastectomy. It's quite common, but rarely is problematic. The problems that we normally encounter after seromas is those patients who will need repeated aspiration, which can lead to wound complication and wound sepsis, and it can also delay in the starting of adjuvant radiotherapy. The etiology is multifactorial and mainly patients with locally advanced breast cancer, high nodal dissection, and increased BMI. Different techniques have been tried to reduce seroma formation, and all of them involve obliterating the dead space. The standard of care is using low suction drain. Other techniques that have been found to reduce seroma is quilting, where the skin flaps are fixed with sutures to the anterior chest wall, and fibrin glue. The same trial compared standard management with fibrin glue and quilting, and the initial results showed that quilting and fibrin glue were superior to the standard of care. And later, the following results showed that quilting was superior of the three techniques. The aim of our study was to compare post-mastectomy seroma drainage between patients who had standard care and those who had fibrin glue applied to their flaps prior to closure. It was a retrospective audit over a period of 1 year, and patients were divided into two groups, those for standard care and those for standard care with fibrin glue. And the drains were removed if the output was less than 50 mils per day, or those who reached day 10 post-surgery. We had complication with COVID that the group 2 patients who had fibrin glue ended up being discharged home with their drains, and the drains output was monitored by the patients, and when the output was less than 50 mils, or when they reach day 10, they will go to a local clinic for removal of the drains. We had a cohort of 66 patients who received 72 mastectomies. Group 1, we had 39 mastectomies, and group 2, 33 mastectomies. Group 1 had a total mean drainage of 352 mils, whereas group 2, which had a mean of 372 mils, which was not statistically significant. However, those patients who had fibrin had an average drainage of about 82 mils compared to 102 mils in the standard group, which was statistically significant. The patients who were discharged home with their drains, however, ended up having their drains left for longer, ending up with a mean average day of 4.6 days compared to those who were managed in hospital having an average of 3.5 days. Overall, there was no difference in the total drainage between the two groups. However, the fibrin group had less drainage per day. As I've mentioned, limitation to our study was due to patients who had the fibrin being managed at home and the drains were left longer, and we couldn't do a cost analysis. When we compare our patients' mean average drainage with other studies, we saw that there was no difference between our group and the patients in the same trial who had glue. The only study that had difference was the study done by Benevento in Italy, where the glue patients had an average of mean of 94 mils compared to those who had standard care having 176 mils mean drainage. But in this group, they mixed patients with mastectomies and those who had breast conserving surgery. In our study, we saw that only about 46 patients had early disease compared to the same trial where the 73% of the patients had early disease. So our patients were more advanced disease compared to the same trial. In conclusion, we feel that intraoperatively use of fibrin is safe in a cohort of women with locally advanced breast cancer. The trend was that the drainage is reduced in the fibrin glue, and we think further studies need to be done to evaluate whether patients who had a fibrin glue will need early removal of drains and if there was any delay to adjuvant therapy. But our impression at the clinic is that we are seeing less and less more of patients who need seromas to be drained compared to previously when we didn't use the fibrin glue. And that's the long and short of my presentation.
Thank you very much, Bernard. I think it's also not the long and short of your study, as you know, that we're going to, Bernard's going to go on and complete his MEd. He's just finished his exams and look at stage. Etienne, I see you put a question in the chat function, which I think Etienne has actually dealt with. He said, thank you for presenting the study. I think an important endpoint is patients presenting with recurrent seroma after removal of the drain. This might require aspiration, operation, interfere with radiation. Did you look at this? So no, we couldn't manage to look at it because of the different variables. Basically, if we had not had COVID, if we had not changed everything in the clinic and the way we ran, then we would have been able to make a better comparative study. Sorry, Bernard, I've answered your question. Did you want to say, did you want to answer Etienne's question in any other way?
No, I think you covered it well.
Okay, all right. So I'm looking forward to Etienne, sorry, not Etienne, Bernard now being able to collect the rest of his data so we can actually look at more details of our patients. So thank you very much. I'm now going to hand over to Francois, who is the head of the breast unit at UCT, and listen to some of the research that they've been doing there. Thank you very much.
Thank you, Jenny. I'm going to introduce Kevin Naidoo. I mean, the Cape Town Breast Forum has asked us to present to the audience a little bit about the research that happens at the two universities. So Jenny has asked a speaker from Tygerberg, and I'm going to introduce Kevin Naidoo to you from UCT. He's been doing research with me actually also in the beginning when we did this initial trial. Jenny was also involved when she was still at Christian Barnard, but it was mainly done at UCT. So just a little bit of background about Kevin. Kevin is not a breast surgeon or an oncologist. He's got a BSc. He's a scientist. He qualified at UCT and then he did a PhD at the University of Michigan and in the US. And he chairs the scientific computing research unit at UCT in the Department of Chemistry. And we've been doing research with him where we're trying to manufacture a liquid or trying to identify a liquid biomarker that can be used in the diagnosis of breast cancer. And I'm going to leave it to him to explain to you how this trial worked. He's basically going to present data from the initial pilot study we did, and we are busy with follow-up studies on this. So thanks, Kevin. Thanks for agreeing to speak to us all, and I'll leave it over to you.
Thank you, Francois.
Perfect.
Got it? Excellent. Okay, thank you, everybody. Sorry about the slow start. I'm going to talk to you a little bit about as Dr. Aleba has been explaining, a study that has been running actually for quite a while. And it started with our initial discovery that the genes that express for the alterations of carbohydrates on proteins and these proteins are glycoproteins, had a classifying capability for distinguishing different types of cancers. So that work was done. Is that slide changed? I think it has. That work was done because if one looks at the existing FDA-approved cancer biomarkers, you'll notice that there's about 70% of that of those are in fact glycoproteins, and some of them are glycolipids as well. The ones for breast cancer, yeah, the CA15-3 is a sort of reduction from a chemical point of view of mucin 1 or MUC1. But all of these, what happens with all of these glycoproteins, the reason why they are biomarkers is because the carbohydrate bit, or the complex carbohydrate bit, has been modified by these glycoenzymes. And so what we decided to do was to look at the glycoenzymes. We took if you like, a big data study of the cancer genome atlas. And what we did was we looked at the differential expression between cancer types and normal tissues as well, but certainly between cancer types. This is what this is showing over here. And we found that there was a differential expression between, for example, breast cancer, colon cancer, ovarian cancer, for this interest of this group. And the glycoenzymes differentially expressed and were either upregulated or downregulated for these different cancer types that allowed you to use these as features to distinguish the cancer types. So we then pulled out one of the cancer types, and because our interest here was breast cancer, and as Francois mentioned, we collaborated across 3 hospitals. Actually, we looked at patients at Groote Hospital, Victoria Hospital, and Christian Barnard, where Jenny was just about leaving out the door as we started the study. And what we did was one of the reasons why we actually went to Christian Barnard is because we started running out of patients at Groote Hospital, because we wanted patients that had not received adjuvant chemotherapy while waiting for surgery, because what we were looking for at the time was to get a section of the tumor and compare it to the section of adjacent normal tissue on the same patient, and then to get a blood sample from that patient. And we also had a cohort of volunteers where we took blood samples from those volunteers, because we attempted to look at blood samples at the time. That wasn't very successful for technical reasons. And so we just focused now, and yeah, I'm just going to show you the proof of concept results, the results of the proof of concept clinical study. So we had 15 female patients. It's a proof of concept study, so it's a small cohort of patients. They were between the ages of 31 and 76 years old. And when we analyzed, we did a full RNA sequence of both the tumor tissue and the adjacent normal tissue, and we found 3,065 genes were differentially expressed and met the statistical measures of the false discovery rate of, I mean, these are just technical statistical terms, of 0.01 and a log fold change, which allowed us to look at, confidently look at these 3,065 genes. Now, when we took the full 3,065 genes, and we did something that's fairly simple to do, which is something called a principal component analysis. It was fairly easy to distinguish between the normal tissue, which is highlighted here in colored in red, and the turquoise colored tumor tissue. And you can see that one could actually just even draw a straight line sort of running through this, except for that one sample over there, which just keep this in mind, it's the GT14 sample, which seemed to, we're not sure why it's sort of showing up on the side. I wouldn't want to blame the surgeon or the pathologist who took the sample and sort of might have mixed it up. So we'll say that the data showing, not clearly showing that sample is on the side of the tumor expression levels, the components. So that's fine to take the 3,065 genes, but the problem is, and the problem generally in gene expression analysis, is that you're dealing with something called the curse of dimensionality. You've got far more features than you actually have samples. So you've got 3,065 genes, you've got 15 patients. This is not very useful if one wants to narrow this down, and also to develop a kit that is practically usable in a clinic. So the issue then is, how does one reduce this sample size? And so one can do different things. You can look at something called the landmark genes, and the landmark genes are a reduced gene set that is often used for various diseases, not only for cancer. And it is used because it expresses, it has a similar expression pattern to about 12,000 genes that are very good indicators for a number of diseases, and in this case, particularly cancer. So when we took the 970 landmark genes that we found in this differentially expressed set of 3,000 genes, we found that these genes were unable to show the clear delineation that we saw when we used all 3,065 genes. However, when we took the 26 in this set here, we only use glycosyl transferase genes, which is basically the glycoenzyme genes, but a certain subset of that. Once again, we found a differential expression, and what we showed that those 26 genes were correlated quite closely to a number of important tumor pathways, oncogenes, and certainly the motility circuits, the viability circuits, and so forth, the so-called hallmarks of cancer. What you see here is the log two-fold change. So these are upregulated, and these ones are differentially downregulated compared to the normal tissue. Just get this to go. Right. So when we try to use a simple principal component analysis on both the landmark genes, the 970 landmark genes, and the 26 glycoenzyme genes, these were totally incapable of showing a differentiation between tumor tissues and normal tissues. What we had done, and since our expertise here is, we do lots of machine learning, and I saw Jenny making a reference to artificial intelligence replacing certain things that is done in the health profession. but this is some of what we do. And so we wrote, we had a large piece of code that we had written, which is something called a denoising autoencoder self-organizing map, fancy name, but basically what it means, it is an unsupervised learning method that is able to discover classes if well constructed. And so this DASA method, we use this DASA method and the 26 genes, and we use the data of the 26 genes, and we found that we were able to get extremely good classification. In fact, we got the same kinds of classification as when using the full 3065 genes. And so what you see here in this display panel here, that we, these are different methods that we're using. And when we're using our method, this DASM, which is a non-linear method, is much more complex than the PCA method, we get 23 out of the 24 samples. So remember, what we're using actually is only 12 patients. So there's only 12 patients that we're using. 3 of the 15 patients, we used that data to develop the primers and all the other, set up the experiments and the benchmarks for the experiments, the genetic part of the experiments. So what you see here is a 23 out of 24 discovery rate, which is exactly what you're getting for the 3065 genes. And in fact, the problematic sample was this GT14 tumor S4, which is the one I pointed out to you, which also came up in when using the full 3065 genes. So this is very promising. This was a proof of concept using a fairly small amount of patients, and this in statistical terms is phenomenal to get this kind of discovery rate with a fairly small gene set. And let me just put that in context. If one were to use the PAM50, that's 50 genes that you're using for differential expression. If one were to use the mama print, that you're using 70 genes in that case. So now we're looking at 26 genes, and it immediately becomes viable then to build a small kit that one can then use for diagnostics purposes and possibly prognostic as well. But if one wanted to be a bit more ambitious, and then the question is, then could we then use the blood samples? And previously we had tried to use the blood samples, but we were limited, in fact, by limited funding. And so we tried to use the same extraction kit and a sequencing kit that we had gotten from Thermo Fisher for the blood samples, as we had done for the tissue samples, and that was just not possible. It wasn't, we weren't able to do that. And on top of that, the other problem was that at that time, extraction of what we were looking then for was cell-free RNA, was not very common and not very possible at the time. And so we were really pushing the boundaries at that point in time. So the question, what we really would want to do then is, if one asks the question, can you use blood samples for detection and early detection and non-invasive detection, you can use a number of these genetic features in the bloodstream. You can use cell-free DNA. There are issues with cell-free DNA that I won't go into right now. There's circulating tumors DNA. There's cell-free RNA, which is the one we're using, which gives us easier access to differential expression. And there's also the circulating tumor cells as well. The reason why blood is a good source of potential early diagnostics is that it holds a lot of these exome genetic material that moves around in the fluid. and it's intimately connected to the effects of immunity and inflammation and stress signaling and cellular communication. And so the question then remains is, can we use the blood glycoenzyme gene expression, and can we link this to clinical and biochemical and pathological changes? And this is then this is where we are at the moment. So we've got, we're undertaking we've passed the proof of principle clinical trial, which we showed that there was in fact a very positive, and this, we haven't published any of this work. The university is looking at how do we actually protect some of this discovery without having these large pharma just take the discovery and run with it. So that's one of the reasons. So in fact, this is probably one of the first sort of public discussions that I'm doing on this. So we've got the proof of principle clinical trial done. We've developed very sophisticated deep learning algorithms that allows us to look at biological materials rather than using standard statistical methods. So this is a big advantage that we've got at the moment. And we have just, at the beginning, actually during COVID, I was successful enough to get a small amount of funding for us to launch the phase 1 clinical trial, which is underway at the moment. What's next is that what we aim to do in that clinical trial is to not only look at the tissue glycoenzyme gene biomarkers and to confirm those genes that we've already discovered, and maybe tweak the expression set more. But we also would like to look at the blood samples as a possible source for biomarker development, with the plan to develop a low-cost glycoenzyme expression device. And I'll mention that, talk a little about that later. So where we are at the moment is we've working with Thermo Fisher, and we've gotten a specially designed panel for us to analyze the blood samples, where we can extract from the exosome this cell-free RNA. And the clinical study led by Francois has started in July, and we have about 22 of the 30 patients. Okay, these are the cancer patients in the study at the moment. We need to get just 8 more to complete the study. The strategy we're using now, and the reason why we're successful in getting the patients far quicker than we had before, is that we are confident enough now to use biopsy methods and use the surgical skill of the team at Groteskia to extract both the samples from the tumor and the adjacent normal tissue, and Francois can talk to you about that. The pathology is also ongoing. So we do a pathology study at the same time. We don't look at the results at all. We look at it afterwards to see if our methods are accurate enough to reveal that. The genetics should start in September, and then we do the bioinformatics and data analytics, which we hope will finish not later than February 2022, but hopefully before that. So that's the long-term. The long-term objective is actually to get to this. So, I mean, it will be great to get a panel, and it'll be great to get to a point where we've got a South African developed panel that will do the same, and hopefully in better than does the PAM50 or the MamaPrint or any one of those biomarkers. genetic-based biomarkers. But what would be really wonderful, and here we've got collaboration that we worked actively with people in electrical engineering, and that is to develop a low-cost device which we can use. These devices that is made by Oxford Nanopore that allows you to do sample collection in a field or in a clinic. And then the combination of that, and in fact, your mobile phone with some clever circuitry can hopefully get us to a point where we can do diagnostics in the future in rural clinics. So that's where we're going with this. The team that we've gotten, and I'd like to, some of them have been part of the proof of concept studies and have moved on, and some of them are still with us. These are my postdocs and students in the front here, Christos Ferlas, who's a Greek postdoc that decided to come to South Africa because he really liked the windsurfing. So it shows you we can attract people, not necessarily only for the science, and the science is an incidental beneficiary of our beautiful country. I work closely with Raj Ramazar and his colleague George Rebello. The CPGR did the proof of concept sequencing, and then the clinical collaborators are we're not working at the moment now with Jennifer Parks, but she's been part of the conversation since the beginning, and then Francois and Lydia Cairncross, and at Christian Barnard, Britta Dedekind did a lot of the sample and patient recruitment along with Jenny Edge. And Dhiren Govender, when he was at UCT, led the pathology studies, but that's now been taken over by Darshni Chetty. And then Jennifer Moodley's working on the epidemiological aspects of that. So with that, thank you very much.
Thanks, Kevin. I think it's just nice for people to see, what exciting works are happening at the universities, and what hopefully we can design something that really we can put to work in our clinics and be to the benefit of our patients. I don't know if there's anyone that wants to ask Kevin some questions. Jenny?
No, thank you. Thanks very much to both of you. And it's very nice, for those of you who don't know, one of the things that Francois and I have been doing is trying to actually bridge the gap between the universities. And we share a common Vula app for referrals, and we're moving in that direction because I do think that united we're much stronger. So thank you very much for this. And Kevin, it's really nice to hear what's happened to your work. Okay, thank you. Francois, shall I continue, or did you want to say something else?
No, that's fine.
Okay, so it just says for me to wrap up the meeting, and there are a number of thanks. First of all, I'd really like to thank our speakers, and I'd like to thank Francois for introducing the UCT side. So thank you very much for that. I'd like to thank all of you for giving up your evening, because clearly without all of you, we wouldn't have the Cape Town Breast Forum, and I think it is a very important forum for us to discuss points, and I think it has been, a really very interesting evening. I'd like to thank Wendy, who she sponsored the meeting. She is from Beatrice, and I've been told to tell you all that was previously Milan, because I was quite confused about who Wendy was working with. But Wendy, thank you. It really is a very worthwhile sponsorship, I feel, and direct sponsorship is hugely helpful. As always, Barbara behind the scenes, pushing us all and making sure that we're all on time and doing all of our bits and pieces. And then the technical team, Jean-Claude and his team from Birgit and Terence, thank you very much. And so all that remains for me to say is, Etienne, would you like to close this meeting and give us a date for the next one? Thank you.
Okay. Yeah, so just, Jenny, thank you very much for running this meeting with your normal exceptional efficiency, and I think it was a really good set of topics. Our next meeting is scheduled for the 8th of November. Just to remind everyone, we sort of have a a collaborative steering committee that set up all these meetings in advance and decide on the different topics. We've identified a meeting chair for each one of these meetings. Our next meeting will be shared by Sarah, I think, if I remember correctly. And we've provisionally also scheduled that topic as being radiotherapy in breast cancer. We'll see if that remains the same. But in principle, the meeting date has been set for the 8th of November. Please keep an eye out on our website, www.CTBCF, Cape Town Breast Cancer Forum.co.za. The dates are on there. Today's schedule was on there, and we will try and have these talks of tonight put onto that website as soon as possible. The previous meetings talks are on there as well. So I'm going to just sort of try and use that website as a bit of a hub so that we can keep everything together. So just keep an eye out for that, but maybe just in your diary, put the 8th of November in there for our next meeting. We'll see what happens with COVID. I think everybody's fairly fed up with webinars and Zoom meetings, but we'll have to see, what the situation is at that stage. So we will communicate with everyone. And then, all for me to say is thank you very much for giving up your time for this meeting, and I hope you have a wonderful week. Enjoy that. Bye.